Hi everyone, welcome. My name is Cameron Lilly. I'm a Scientific Affairs Liaison with Menarini Silicon Biosystems. What I'd like to talk with you about today is something that's plaguing molecular pathology, and that is failures as a result of paucicellular samples, or low cellularity. What we're going to do today is talk first about the technology that we're going to leverage to try to address this problem, and that is the DEPArray digital cell sorter. We'll talk about the instrument, and then how cells are handled and identified and then isolated using this technology. Then we'll talk about the problem itself, and that is the problems that arise from low cellularity samples with FFPE specimens in the modern era of pathology. Then we'll tie it all together by talking about how we can attack these paucicellular samples using the DEPArray to get single cell copy number variation analysis and pooled cell next generation sequencing. So what is the DEPArray? The DEPArray is a benchtop digital cell sorter designed to isolate rare single and pooled cells with 100% purity. It does this by combining immunofluorescent identification and bright field microscopy to identify cells, and then uses precision semiconductors and dielectrophoresis to isolate and move those cells for recovery. This is all managed with the cell browser software, which collects the fluorescent data and enables either automatic or operator-designed selection of the cells. It captures high-definition images. Some of the platforms are extremely automatic, including the FFPE, and it's a high-performance and user-friendly instrument to use. One of the big strengths of the DEPArray is the high-confidence cell identification. With the multiparametric selection of fluorescent markers, either 5 or 9 fluorescent channels, this allows for a broad characterization. The inclusion of DAPI allows us to determine if cells are nucleated or non-nucleated, and the bright field microscopy allows us to do phenotypic identification as well. How big are the cells? Are they symmetrical? And what shape are they? And even are they centered within the DEP cages? We'll talk more about the DEP cages in a little bit. This is an example of how we can use the multiparametric analysis on the DEPArray and the identification and data captured from those cells to determine which cells we would want to then collect and route if we were looking for single or pure pool cells. What we have here are 4 rows of images that each correspond to a single object or region of interest within the DEPArray instrument, and as we move from left to right, first on the left we have the PE channel, we have the DAPI channel second, and third we see the APC channel, and then we have the bright field. The 4 on the right are combinations of thereof in order to help us get an idea of what our combined images might look like for these samples. Our top row, it looks like we do have a cell there. We see DAPI signal and stains with PE. We see, though, an unlocalized APC positive sort of blob on the outside, indicating we might have more than one cell there. It's also DAPI positive, so we wouldn't want to isolate that one. Similarly, in the second row, we see PE and DAPI signal, but the APC signal isn't strong. It's localized to a fleck on the surface, probably not ideal either. In our third row, again, we have PE and DAPI signal, but no APC signal that looks like background. And there's a dark spot on the outside, maybe a membrane bleb, so it's possibly debris. We wouldn't want to isolate this cell either. However, if we look at our bottom row, this would be ideal. We see strong signals in all of our channels. It's a nice round cell. It's by itself, so this gives us the power, combining fluorescence and bright field microscopy, to isolate single cells and either isolate them as one or pool them together as a new sort of subsample. Cells are handled in the DEPArray on the cartridge, which is pictured here on the left. This is a self-contained, single-use cartridge into which your sample and buffer are loaded and then placed into the instrument. Once it's in the instrument, the buffer and sample are flown into the main chamber using the contained microfluidics, and then charge is applied to the small semiconductor array, and the cells are separated and held in place using tiny electrical charges by a process called dielectrophoresis. We'll talk a little bit more about that in a second. Once the cells are in the main chamber, though, this is where the image data is collected by the onboard fluorescent and bright field microscope. This is done during a process called chip scan, in which each of the fluorescent channels and bright field images are captured, and cells are identified within the software and given a unique identifier. To that unique identifier are attached all of the fluorescent and bright field data, so all of this can then be analyzed when you want to isolate the cells which meet certain criteria which you can set. Trapping and moving cells within the main chamber is done with what are called DEP cages. A DEP cage is not a physical thing. What it is is a series of semiconductor electrodes with small charges applied. You can see a diagram of what this might look like on the right hand of the slide, and in the right box is what we would call a DEP cage. It's a single negative electrode surrounded by positive electrodes. That negative electrode represents an area of least resistance, and that's where the cell will naturally move to. It's very gentle, it doesn't put any physical pressure onto the cells, and because of the properties of dielectrophoresis, the cell actually doesn't even sit on the bottom of the chamber. It actually hovers between the bottom and top of the chamber and is held there by this dielectrophoretic charge. Now, to move a cell using DEP cages, all we have to do is change which of the electrodes has the negative charge under it. So if we wanted to move a cell in our highlighted DEP cage to the right, to the green area as a highway to route that cell, we would simply stepwise move that single electrode with the negative charge one by one, and that would carry the cell along with it. And that is how we can trap and move cells within the DEPArray to isolate cells very gently and to get exact single cells and exactly the cells we want every time. So how can we use this powerful technology for cell isolation? Well, one way is to rescue Paucicellular FFPE samples utilizing the DEPArray technology. So why is this such a big problem? One of the big key things that is a growing arm of modern molecular pathology is to collect next-generation sequencing data from FFPE prepared specimens from patients. This mutational analysis is critical. It can inform treatment decisions, but it also informs our understanding of cancer. But one of the big issues here is that sample quality remains a massive hurdle in trying to do these analyses. One of the big challenges is that formalin fixation, paraffin embedding, and the reversal of those processes is inherently destructive to nucleic acids. Tumor cellularity is an enormous part of this as well. Over 80% of the failures in this molecular analysis come from the fact that the sample has just too low percentage of tumor cells to get you meaningful information about the genetic aberrations in the cancer. As we talked about the inherent destructive problems with FFPE preparation and deparaffinization, decross-linking, recoverable DNA content and quality is also very difficult and needs to be monitored and can lead to failures too. But the FFPE sample testing rate failure for this is about 8% or greater, and what this means is tens of thousands of samples a year cannot then be analyzed by these molecular methods. They would fail this analysis, and the single biggest driver of that is low tumor cellularity or paucicellularity. However, using the DEPArray, we've developed an FFPE workflow to take FFPE specimens, prepare the samples, sort those on the DEPArray, get stromal cells or non-tumor cells for background, get the tumor cells to do library preparation and sequencing to get molecular data out of these samples. We published a methods paper on this in 2016. The citation is here for your information. And I do want to highlight on the next slide one of the important figures from this paper. Shown here is a table of the sample types that were used here. The specifics of exactly the kinds of samples are not necessarily important for you to take home, because I'm going to walk you through some of the big take-home messages. One of those is that the tumor types were broad. We were able to analyze tumor tissue from a number of different kinds of tumor types. And in our first red box here, I want to highlight this particular sample in that we successfully did analyze a sample with a tumor cellularity of just 5%. That means that just 5% of the cells within the tumor sample were the actual cancer tissue, but we were able to get meaningful information out of this. So we're able to go well beyond the limits of other methods, and a sample like this wouldn't even be considered for the standard molecular methodologies. Our next set of red boxes highlights the storage time in years of some of these samples. We were able to use this method to analyze samples that were as young as 1 year old, or as old as 21 years. So these samples had been isolated and stored for over 2 decades, and we were still able to get genetic information out of these. And so what that means is that if we move on to the next image, and we have a pathology slice that looks like this, with our very, very small islands of tumor cells in red circles here, This means that this sample, which would otherwise have to be discarded and not considered for molecular analysis or would fail if it were pursued, now we have a method for getting at the information within those tumor cells and the stroma from within this sample. So what does this process look like? Well, the first step is to prepare the specimen by disaggregating the tissue, staining it, and checking the quality of the nucleic acid. So, first step is to take a tissue slice from your FFPE tissue, deparaffinize it, and equilibrate it into an aqueous buffer. Next, the sample is disaggregated so that we can get a single cell suspension for the next step, which is staining. Now, our kit, the DEPArray FFPE Sample Prep Kit, labels cells with anti-keratin, anti-vimentin, and DAPI. We'll talk about the purposes of those in a second. However, this platform is technically open and you can stain with other markers, and we'll see an example of that at the end of the talk. The next and very important step here is to take a small aliquot and run a PCR-based quality control step to determine the quality of the DNA recovered from your sample. This is done before DEPArray sorting in order to determine if the molecular quality of your DNA template is good enough to even pursue sorting the sample. So, what are the kinds of tissue types that you can put into our kit? Well, we tested keratin-positive carcinomas and we validated this in lung, breast, pancreatic cancer, colorectal cancer, ovarian cancer, and prostate FFPE sections. There is an important critical step here, and that is the section thickness. The thickness of the section before going into this sample preparation needs to be between 20 and 50 microns in order to recover whole intact cells. The process of dielectrophoresis relies on intact cells and intact membranes, so we have to have a thicker section. Okay, so we've got a stained, disaggregated, single cell suspension from an FFPE sample. Now we need to sort it on the DEPArray. So, the cell suspension is added to the DEPArray cartridge. This is then placed into the instrument. The cells will be pushed into the main chamber, imaged, the fluorescence data captured, and we can use the cell browser software to group the cells however we want and then isolate them. For example, and you'll see this a lot in the chat today, is stromal cells, so healthier baseline cells, compared to our tumor cells. That gives us an internal control and compares it against the diseased tissue. And we can isolate those cells either as single cells or as pools of cells, depending on what kind of analysis we want to run downstream. This is an example of what this actually looks like on the instrument. This will seem fairly familiar if you're used to running flow cytometry, and we are looking at an XY scatter plot with two fluorescent parameters. Across the bottom on the x-axis, we have keratin staining with Alexa 488 labeled antibody, and on the y-axis, we have vimentin staining with Alexa 647. Each of these dots represent a cell, and they're given basically a intensity score based on their fluorescent parameters. Our vimentin Alexa 647 antibody will label our stromal cells. These would be our healthy stromal cells that we would want to keep for control. Our keratin will stain our tumor cells. Our double positive cells will be cells that are undergoing EMT, and our double negative cells could be tumor infiltrating lymphocytes, stem cells, or other cells within the biopsy sample. The inclusion of DAPI in this analysis is twofold. One is to make sure that the instrument can reliably identify every cell, but what's also important is that by identifying the stromal cells and looking at the DAPI intensity of those normal healthy cells, we can actually identify tumor cells by comparing their DAPI signal intensity to the stromal cells. They'll have amplifications and deletions and aberrant amounts of DNA, and we can actually detect that using the integral intensity of the DAPI signal in our tumor population. So this really gives us the way to unlock the potential of FFPE samples. We can do mutational analysis and next-gen sequencing on pools of cells. We do copy number analysis on pools or even single cells from FFPE tissues, and that gives us the ability to look at heterogeneity within FFPE prepared specimens. So the next step then would be downstream analysis. We need to lyse the cells and do a library preparation. This is an open process. You can use whatever library prep method you want. We do offer one in the Ample One kit line, but let's talk about that. So what are the limits here? How far can we push this? As I alluded to, because we can sort the tumor cells and compare them directly to the stromal cells from the same specimen of tissue, at this point, tumor cellularity is now not really a requirement for successful analysis using molecular methods because we can do this isolation and enrichment. With this workflow, if you wanted to do targeted sequencing using NGS, you could do small pure pools of cells between 100 and 300 cells, or if you need a more high-level analysis, but you want to do single-cell analysis, that can be done with low-pass genome sequencing downstream of the DEPArray workflow as well. This is, of course, contingent on getting good quality DNA, but if the DNA is there and your QC score is good, you can absolutely take just about any FFPE sample into this methodology. So this diagram represents the power of pure pools of cells. On top, this represents a copy number variation readout, or low-pass genome sequencing, of an unsorted specimen. So this would be, take a sample of it, sort, or do CNV analysis on all of it. As you can see, we do see some red, some blue indicating areas of amplification and deletion, but it's noisy, doesn't seem like there's a strong baseline. What happens if we then take the same sample, sort, analyze the stromal populations and the tumor populations separately? Well, for one, our stromal baseline is now nice and uniform. We have a really good background to compare our tumor tissue to. And most importantly, in the bottom, if we now look at just the tumor cells, when they are no longer being diluted out by the stromal background tissue, now we have a tremendous insight into the genetic aberrations in these tumor cells that we could not see before because they were being drowned out by the background healthy cells. So now we have an accurate CNV report for what's going on in these tumor cells, which we simply couldn't get without this kind of isolation. So that was on pools of cells. What about if we want to look at this at the single cell level? What we did here was take FFPE biopsies, run them through our kit preparation, and then do WGA and low-pass sequencing using our AMPLA1 kits. And what we found is that when we looked at a range of numbers of cells from 117 down all the way to 1, is that using this methodology and doing FFPE analysis on single cells is absolutely possible, as the CNV profile in this sample did not change irrespective of the number of cells that we were looking for in this analysis. The CNV profile for 117 cells is the same for 30, for 6, and for just a single cell out of this FFPE specimen. So we have really tremendous and unparalleled ability to get genetic information out of FFPE specimens. What does this look like in a real-world example? Well, here's a good example of how this can be used to find extremely rare malignant cells in classical Hodgkin's lymphoma. And this is the isolation and characterization of a type of cell called a Hodgkin and a Reed-Sternberg cell. These are very, very rare cells within this particular cancer, and they're very hard to get. So trying to understand the mutations that are driving it by a bulk method in which you're analyzing a sample all at once, you're going to have that same background stromal dilution problem. However, we can use the DEPArray to isolate these cells and get CNV profiles of the healthy tissue and of the extremely rare HRS cells. Now, I mentioned that the DEPArray FFPE application is open in that you can do staining aside from the staining that is included in our FFPE sample prep kit. And that was what was done in this particular case, as you can see in the top left. We're looking at DAPI signal and CD30 signal and PDL1 signal. These are markers for these HRS cells. Once we have isolated each of these different kinds of cells using the DEPArray, we then can look at the CNV for bulk, or we can look at the CNV for the sorted leukocytes and of these sorted HRS cells. And what we find when we do this analysis is that the bulk cell analysis really doesn't show us any copy number variations looking in panel D. If we compare panel E, and this is the purified leukocytes from this sample, we see a normal profile. This is what we would expect. However, now that we have isolated pure HRS cells, we now are able to detect the copy number variations in these critical rare cells which are malignant in this disease, and now we can make accurate deductions about what the genetic aberrations are within these HRS cells that we simply were not able to do without isolation on the DEPArray first. So, the DEPArray platform rescues irreplaceable low cellularity FFPE samples and empowers your preferred molecular analysis.